as we go. Welcome to week 4. We have made it to the end of this course, which is very exciting. I feel like we have learned a lot and worked through a lot together, so I'm excited to kind of tie it up today. And so this week's topic was actionability, but we'll also kind of open it up to all questions from all 4 weeks and then talk about the final quiz. So our agenda, the question and answer session grouped by topic from the Google form, then we'll review the quiz questions, and then live Q&A from the content from all 4 weeks. So you feel free to ask questions that you have that have come up across all of the weeks if we have time, which I'm hoping we'll still have a little bit of time at the end. So, oops, hold on, where is this tiny URL here? Here we are. Sorry about that. I'm going to move this to the front. Accidentally stuck all the questions in front of it. Okay, so one last reminder that completing this quiz, which is only 5 questions, is required to complete this course and receive your certificate. It will not be graded. We're going to talk about the answers today. I think I have 91 responses at this point. So if you've not done so, please complete this quiz now. That's why I am putting the answers in the middle of this session. I made a tiny URL so it's easier to type in, tinyurl.com/papery quiz. I'm going to try to put this in the chat. Okay, that's now in the chat. So if you have not taken this quiz, please take the quiz. We have 91 people who have taken it so far. Thank you. Okay. So let's start with our questions from the week. And they're grouped by topic again. So I had some questions about uncertainty of your variant of interest versus the actionability of the condition. And something I will say before I get into these questions is that the actionability working group, when they're performing their assertions and their scoring, is assuming that you have a likely pathogenic or a pathogenic variant in the gene that they are evaluating. So that's something that's really important. While a gene condition pair might be highly actionable, that has not necessarily anything to do with the variant. So if you have a VUS, you still have a VUS. They are assuming that you have gotten to likely pathogenic or pathogenic in most cases for your variant curation to be able to act on the actionability, if that makes sense. So the question here is, in lower and middle income country settings, when actionability evidence exists, but local feasibility is limited and variant assertions conflict, how does ClinGen recommend scoring actionability and reporting uncertainty to clinicians and families? So as I said, in general, actionability is at a gene condition intervention level. ClinGen's actionability groups may mention certain variants, but that is usually common causes of disease. And we know that this is in ascertained or published populations, which can be biased. Please mute if you are not muted right now or getting a lot of background noise, or Wafa, if you could mute, thank you. So we know that these common causes of disease in ascertained populations are biased. So I wouldn't necessarily expect your variant of interest, even if it's a common cause of disease in an LMIC that is not represented in the literature well, to be listed in an actionability report. So it is up to the testing laboratory to classify each variant to determine the level of evidence for that variant. If the variant is a secondary findings add-on to a diagnostic test, only likely pathogenic and pathogenic variants should be returned for this reason. We don't want people to start to act on variants that are uncertain, because we've seen in our variant classification week that there are different tiers of uncertain significance variants. They could just be variants that stay uncertain forever. They could be variants that bump down to likely benign. They could be variants that bump up to likely pathogenic, depending on the amount of evidence in that VUS bucket. So in cases with VUS variants for affected patients, which actionability is usually for those secondary or incidental findings, but if you have a VUS variant in an actionable condition for an affected patient, it's often more useful, and we've said this in previous weeks, to act on and treat their current disease diagnosis than to make medical choices based on that VUS variant, unless they may include non-invasive screening. If it's really just upping screening protocols and they're not invasive and they're effective, it might make sense to potentially add some more screening based on that VUS. But you wouldn't necessarily want to have surgical interventions or highly invasive interventions based on that VUS variant. Okay, so another question about actionability versus variant curation. How should I evaluate and decide whether to report a potentially actionable variant when it is determined or identified from a sample with low DNA input and low exome sequencing read depth? So in general, you in a laboratory, you should set quality thresholds for all of your tests. So in our lab, we have decided to confirm variants using alternative technology. So we'll do exome or genome sequencing. And if we find a variant that we question the coverage of this variant, we question whether it might be real, even though it would be a potentially very actionable variant, we need to be sure that that variant is in fact real in the patient of interest before returning it, because that could be extremely harmful to return a variant that is not actually real. So in general, we use confirmation technology. So Sanger sequencing, PCR, arrays, just a different type of technology if the coverage is low or the quality of the variant is dubious. This is particularly critical in a screening setting because you really want to make sure that a variant is real before telling someone that they have a likely pathogenic variant and encouraging them to pursue invasive screening measures, for example, or surgeries, risk reducing surgeries, if we're not sure that the variant is real. So it's very dangerous for a lab to report out a variant if they are not sure it's real. So if your laboratory doesn't have other technologies, we also just suggest that you perform the same technology again and see if you get the variant again. And in that case, if you have low yield, low DNA input, if possible, consider asking for an additional sample. If it was blood and that's highly invasive, maybe you have a baby, you could consider a different type of sample. So we have had some options where it was originally a blood sample and they said the patient is too sick to collect another blood sample. You could collect a saliva sample. You could try to collect a buccal sample, by scraping the cheek. When I say a dubious variant, I mean a variant that you're not sure if it's real. It might be in a region that is highly homologous. It might be in a region that's very difficult to sequence. Maybe there's a pseudogene, maybe it's a really repetitive region. So that's just what we mean when we say that the quality of a variant is dubious. Okay, another question about some of these questions now are about the framework, and I will give the caveat. I myself am a boarded laboratory geneticist, so I perform extensive amounts of gene and variant curation. I have been around actionability curation for 10 to 11 years now as I've been involved in ClinGen. And I've actually provided expert feedback to the actionability group when they've done the hearing loss intervention pairs, but I've never performed actionability curation myself. So these are me answering these to the best of my ability based on working with the actionability group. But as you can tell, it's a bit of a complex process, and they have a lot of very detailed experts in that group that discuss. So these are my best answers here. So for the questions of actionability curation, what is the difference between a score and an assertion? So scores refer to either the final score or the individual semi-quantitative scores that are given for each section of the framework. So for example, a 2 for severity, or for likelihood, a 3C. I was just pulling a curation to grab these scores. An effectiveness score of a T2D, nature of intervention 3, a total score of 10 CD. Those are the scores, whereas the assertion refers to the overall result of the actionability scoring, which can often have expert judgment and consensus in it. And the most recent update of the actionability framework, which was the second paper that I gave you, provides assertion strength that are similar to gene disease relationships. So moderate actionability, strong actionability, limited actionability. So that's what we mean by assertions versus scores. So the next question is, when the four clinical actionability criteria yield discordant results, how do expert panels determine the final actionability score? So you'll see on the scoring website, and this is talked about in the more recent paper. So the second paper I gave you with the strength of the assertions, limited, strong, definitive, you'll see that on the scoring website, there's a suggested assertion and a consensus assertion. So when there are discordant results, the experts extensively discuss and vote to come to a consensus assertion. And so talking about their methods in their manuscript, which was the second one I gave you. They say the resulting rubric has a 4-point scale, limited, moderate, strong, and definitive, and uses the highest scoring outcome intervention pair of each condition pair to generate a preliminary assertion. So with the highest scoring one, they might have a suggested assertion of strong. And you'll see this on the website often. And then it will say like suggested assertion. And then during the actionability working group discussions, predefined criteria and factors guide the discussion, and it produces a consensus assertion for a gene condition pair, which may differ from the preliminary assertion. So you'll see the suggested assertion of strong actionability, and then oftentimes you'll see the consensus assertion of moderate actionability. And I will also point out that in their manuscript, they also mentioned that the suggested assertion, they've never allowed definitive to be a suggested assertion. That always has to be arrived at by consensus. So the highest that will ever be suggested by the suggested assertion based on scores alone is strong. But the experts, based on the data, could bump that to definitive. Okay, next question related to curation. What criteria are used to determine whether a genetic condition is clinically actionable? So there are 4 criteria used to score actionability. The first one is severity of the outcome, or for short, they call it severity. The second one is likelihood of the disease. That they, for short, will call that likelihood. The third one is effectiveness of the intervention. And for that one, they just say intervention. And fourth, the nature of the intervention, or sorry, effectiveness. And then fourth, nature of the intervention in terms of burden and risk to the patient, which is just intervention often for short. And then what criteria does the group use to prioritize which gene condition pairs to assess first? So the actionability working group does require that a gene disease relationship be curated by a gene curation expert panel first and receive a strong or a definitive. They often will not look at any interventions or gene condition pairs that are not curated first by a GSEP. So if they have one that they'd like to prioritize, they will first go to a gene curation expert panel and ask that it be curated if it has not been curated. They will also prioritize genes on lists by clinical practice guidelines, professional bodies. So, for example, the American College of Medical Genetics and Genomics secondary findings list, or genes that are common causes of disease. However, this is based on the published literature, which is biased, which we've talked about. But often these are things that are written about in gene reviews. They will also accept suggestions or nominations from the community, and we'll talk about that a little bit more too. The next question about the curation framework is, how do you reconcile high intervention effectiveness, which is a score of 3, with poor evidence quality, which is a level D? So this is specifically addressed in the scoring challenges section of the original paper. I'm quoting it here. So the protocol developed by the Actionability Working Group relied heavily on published practice guidelines with priority given to guidance based on evidence review. And so the thing to remember is that the actionability working group, much like the expert panels, is really looking for that published or publicly available evidence and not just practice guidelines saying that something is effective. So as maybe you've seen, paradoxically, many published guidelines do not provide evidence summaries for their recommendations. So for those, the actual effectiveness of a recommended intervention is sometimes unclear or unknown. And they provide the example of Fabry disease and recommending that they take aspirin or platelet inhibiting agents to reduce their risk of stroke. But the effectiveness of this intervention was not described in that original source that they cite. So in general, in the actionability working group, they're not comfortable scoring the effectiveness of recommendations without evidence of the actual effectiveness for the patients. And the absence of the supporting evidence really limits the ability to measure effectiveness. So in general, it would be very unlikely to have a high intervention effectiveness score 3 with a poor evidence quality level D. I've most commonly seen when I've just been clicking through the actionability curations a 0D. And to remind everyone, this is what we're talking about here. So you'll see scores for each one of these, which for the different categories, and this is from the first publication, by the way, I didn't pull this from anything else. You'll see scores for all of the different 4 domains, and it has potentially a different definition for each domain. And then letters, which are actually common across all of the domains of the state of the knowledge base. So what's the level of evidence? So this is what we're talking about here. Effectiveness of specific interventions. How effective is the selected specific intervention for preventing or significantly diminishing the risk or harm? And the question was, if you have a highly effective intervention, can you have poor evidence or evidence not provided in the report? And I think they're saying in their publication, the experts in the actionability working group really do not feel comfortable pairing that together, that they need to be able to see evidence to score that something is highly effective. So in most cases, you would see a 0D, which is a controversial or unknown effectiveness with poor evidence or evidence not provided in the report. Because consistent with many of the other expert panels in ClinGen, they need evidence, published evidence, to be able to evaluate on their own to determine a highly effective intervention. Okay, so then I had some questions about representation and really equal representation of genomic data and actionability curations. So since most genomic data comes from people of European ancestry, what specific measures is ClinGen taking to ensure that clinical actionability scores are accurate for populations that are underrepresented or from different ancestral backgrounds? So, as I've said throughout this, we recognize that this is a problem and this is a concern. But as for gene and variant curation, ClinGen just does not generate data. So, we really rely on the community to generate and provide us data to help in a few ways. So actionability will take nominations and they'll take requests for gene or intervention pairs from the community. So if you're really noticing that in your local area or in your community, there's a big increase in publications for a particular gene or a particular condition, you should reach out to the actionability group and nominate that gene and the condition related to that so that they can work on this curation. However, as we just talked about with scores and with publicly available evidence, they do need publicly available data. This is often publications, but perhaps there could be a preprint or some other way that the data are publicly available to be able to perform this curation. So we are happy to work with all countries and all researchers, but we do not generate the data ourselves. We do recognize that the data are biased, and so we help as much as possible to facilitate these curations. And I have another slide on that, I think, or other slides as we go. So another one was questioning representation. So for conditions with low research representation, which can limit the strength of actionability assertions, what support or resources does ClinGen provide for individuals or groups from these underrepresented populations who wish to investigate and help address these evidence gaps? It's a great question. We recognize that this is a big issue. As mentioned on the previous page, ClinGen does not generate data, so we can't generate the data. We also are a largely volunteer funded resource, so we really don't have the ability to fund any of this research. However, I think our biggest strength relies in our size and the ability to support collaborations and foster connections. So I think a really big thing that we can help with is we can support creating connections in disease specific spaces to foster collaboration, which may foster funding or even the ability to have access to technology you might not have originally had access to, by encouraging experts in Africa and elsewhere to join ClinGen expert panels and really start to get to know the rest of the international community in those disease areas in hopes that they can actively collaborate and help each other. We also are trying to give everyone the knowledge and empower them to, work with ClinGen to curate or even curate in their own areas by hosting these training workshops, remotely when we can't travel, and also entirely free of charge, so that we can really work to all collaborate together and educate each other so that we can try to move the needle on these conditions and increase research representation. Okay, so let's move to the questions from all weeks. I had one about variant curation. So the quiz asks about types of variants. So I think related to that is one question of what criteria distinguish a missense variant from other types of variants in terms of pathogenicity assessment. So there are some codes that are most often used for missense variants. However, I will say they're not entirely used specifically just for missense variants in most cases. But when I think of a missense variant, these are the types of codes that I'm thinking to look at right away that are specific to missense variants in most cases. So PP3, BP4, I should say, is the benign code on this side, is computational predictions. So PP3 is computational predictors predict a damaging effect. BP4, I didn't put on this slide, but it's the benign side, are computational predictors predict a benign effect. And so the computational predictors you most commonly think of are those that predict the damage of a missense variant. That being said, there are also splice predictions. So it's not entirely just reserved for missense variant, but those are the ones that I think of for missense variants. Another one I think of for missense variants is PM5, which is a pathogenic moderate code, which means that you found a variant in the same codon, but it's a different protein change. That's often used here. And that different protein change has been previously classified as pathogenic or likely pathogenic, suggesting that that residue might be critical for function of the protein. So that's another one I think of for missense changes. That being said, expert panels have often adapted this now to splicing changes, but this was originally written for just missense variants. Along those same lines, I think of PS1, which is a strong piece of pathogenic evidence that there is the same protein change, but it's from a different nucleotide change. So let's say your variant was a C to T, and then there's another variant that's a C to A, and it's the same protein change, and it's classified as likely pathogenic or pathogenic. It's highly likely that your variant that has the same protein consequence would also be likely pathogenic or pathogenic. So that's a strong piece of pathogenic evidence, most often used for missense variants, but now often can be extended to splice variants. And then one that is largely just used for missense variants is PP2, which is a supporting piece of pathogenic evidence, which is missense constraint at the gene level. And this is looking at missense constraint. That's a great point. I just saw in the chat PM1. I probably should have just looked at the grid. These are the ones that were off the top of my head. But PM1 is another good one that's used for missense variants. That being said, it could also be for, small deletions in a particular region. I guess splicing could end up being in an exon boundary, like in a critical region. So PM1 is a hotspot region. So like a critical functional domain in a particular protein. It's a moderate piece of pathogenic evidence. So that's another good one for missense variants, but it could also be used for some other types of small variants. Okay, we have a couple questions about gene curation. So in resource limited settings, how should diagnostic laboratories balance strict ClinGen classifications with clinical urgency when evidence for gene disease associations is still emerging? So I will say in general, even though the American College of Medical Genetics and Genomics suggests that you use ClinGen curation frameworks, there's no requirement that a gene be classified by ClinGen or that labs exactly use the ClinGen framework when curating gene disease relationships. Many of the clinical testing labs that I collaborate with or work with have slightly adjusted frameworks that can be performed more rapidly than a ClinGen one. However, I cannot say this enough. Gene curation should never be skipped because of clinical urgency. The risk of returning a result that is not in a valid disease gene is too high. You do not want to return a damaging variant that you have not curated the gene just because you have a sick child. Just perform the gene curation more rapidly. The worst thing that you can do is try to act on a nonsense variant in a gene that has no gene disease relationship because you see that it's a damaging variant. So please do try to perform that gene curation. Also, you can refer to the Gene Curation Coalition because it has many more curations, 22,000 or so right now, than ClinGen, which is maybe 4,000 or so. And it may have something about the gene of interest. But really, you need to perform due diligence in the laboratory and at least try to rapidly evaluate a gene disease relationship, or even looking at your data in internal cohorts, because it can be very damaging to return a result that actually has no gene disease relationship just because you're in a rush because a patient is sick. Another question about gene curation is, what practical pathways exist for laboratories in Africa and other underrepresented regions to contribute high quality gene and variant evidence to ClinGen in an ethical and scalable manner? And this kind of also relates to that actionability question I had earlier of how can laboratories in Africa or other underrepresented regions move the needle as far as actionability, and get the actionability group the data that they need to be able to classify actionable conditions that are underrepresented. So there are multiple ways that labs in Africa or other regions can contribute. You could join ClinGen, as I've said, and collaborate in expert panels to be able to share your data while the other experts are also sharing their data. You could submit to ClinVar to share your data if you're able. I can also help with this. I've facilitated many ClinVar submissions. You could publish your data if you're able, and then request that this data be curated and send along the publications. That happens. We accept requests for all of our curation types. So actionability, gene, variant, and even dosage curation. You could become part of a federated matchmaker network so that data can be secure and can stay in your country, but can be shared so that you can really gather that evidence from across the world. And I can also help with this, maybe not potentially directly, but I can connect you to the right people to get you set up to share your data in a secure way in this way, if this is something that you're interested in. Okay, so we're going to do the quiz questions now. Let me check and see how many people. Okay, so we have 111 responses. So this is bumped up. So hopefully, and 112. So hopefully everyone has answered the quiz who is on this call. I put the URL in the chat. And now we're going to move to the quiz questions and their answers. Okay, so these are 5 questions, and I tried to speak to something we learned about every week in our entire workshop. And I would say everyone did an amazing job. The screenshots that I have here of the pie charts of answers are based on like the high 80s to low 90s response rate that I had when I started. But I'm sure that you are did great with your answers here, those people who sent them along. Okay, so what's the difference between ClinGen and ClinVar? ClinGen is a database and ClinVar is an expert curation resource. ClinGen is an expert curation resource and ClinVar is a database, and there is no difference. This is a common question that we get, and I think we talked about this in the first week. So the answer is ClinGen is an expert curation resource and ClinVar is a database. So ClinVar is a database of assertions of variants where you can submit to it. And ClinGen is the expert curation resource that has expert panels that will curate genes, variants, and actionability. And you can see that 94% of folks got this correct. So great job. This was when we only had 88 responses. The next question, a laboratory is evaluating this variant listed here, a proline to a leucine at residue 369. What kind of variant is this? And in some of these questions, I tried to write them in a board style that the answer that you need to provide is the best possible answer. So yes, you could potentially do functional data and determine that this ended up with a loss of function effect. But just looking at the HGVS nomenclature, what type of variant is this? A nonsense variant, a synonymous variant, a missense variant, or you just don't know? And so in this case, if you just look at the HDVS nomenclature, this is a missense variant where one protein change is switched to another protein change. So a proline is switched to a leucine at 369. And you can see that 96%, 97% of folks got that correct in the orange. And I will say, if you're putting these answers in the chat and you didn't fill out the quiz, you need to fill out the quiz because putting the answers in the chat is not sufficient in this case. So I put the URL in the chat. I can put it in again. Please fill that out if you haven't. We have 111 responses right now, so hopefully you've all filled it out. Okay, so the next question, a technical standard, and this is an important question that the expert panels think about all the time for gene curation. So a technical standard publication from the American College of Medical Genetics and Genomics, ACMG, states that gene disease relationships must reach what minimal level of clinical validity? So what's the lowest strength of a gene disease relationship you can have using the ClinGen framework to return variants in a diagnostic setting, so diagnostic testing report? Limited, moderate, strong, or definitive? And it is moderate. I would say there was a bit more confusion about this one, and people tended to go a little higher than that when they were guessing. So we had 77% of folks saying moderate, and a decent chunk guessed higher than that. So a decent chunk said strong or definitive, which is correct in a screening setting. They suggest that you actually go higher than that in a screening setting of healthy individuals that you're testing as strong or definitive. But in a diagnostic setting, when individuals are affected with a condition, they do say that moderate is the minimal level. And so that's why when expert panels are thinking about whether a gene disease relationship should be limited or should be moderate, they really need to ask the question, should variants in this gene be returned on a clinical testing report? If the answer is yes, it needs to score at moderate. If the answer is no, it should be at limited. And that's an important distinction. Okay, so the next question, and this is another one that you have to ask yourself what the best answer is. There may be multiple answers, but what is the best answer? So genome sequencing was performed on a patient with a rare genetic disorder for which no known genetic cause had been identified. A de novo mutation is found in a gene without a disease association. So this gene does not have a gene disease relationship. And insufficient evidence is available to conclude causality for the patient's phenotype. So there, you really just don't know what this gene is doing. So which of the following is the most appropriate next step to build evidence for the identified gene and variant? A, look for incidental findings that could modify the phenotype. B, submit the gene to a genomic matchmaker database to find other cases with the disorder due to the same gene. Or C, use computational algorithms to predict whether the variant affects function of the protein. So for this one, B is the best answer here. About 89% of you got this answer, whereas about 10% used computational algorithms. I think that the critical thing to think about here, and this kind of speaks to the question that we had before, if I have a variant, but there's no gene disease relationship, should I return it if a patient is really sick or if it's a rapid clinical situation? The important distinction here is we have said that no known or the gene is without a disease association. So really the only way that you can continue to get both gene and variant level evidence is to find more cases to have a gene disease relationship that actually makes it to moderate to warrant returning your variant on a diagnostic testing report. So really the best way to do that is to try to gather more evidence to get to a gene disease relationship. While you could use computational algorithms to predict whether the variant affects function of the protein, that's speaking to the variant, not necessarily speaking to the gene and its gene level evidence. So this is why answer B is the most correct answer in this case. Okay, and the last question. According to the American College of Medical Genetics and Genomics, when should secondary findings results be returned? A, when patients who are receiving diagnostic testing consent to receive them. B, to all patients who are receiving diagnostic testing. Or C, to healthy individuals in a screening setting. And so in this case, the correct answer is for consented patients who are receiving diagnostic testing. They often treat this as an opt-in, opt-out model, that you have to actively say you want to receive these additional secondary findings results when a genetic counselor or a clinician is consenting you for your diagnostic testing. And so about 90% of folks got that correct, whereas 10% said to all patients who are receiving diagnostic testing. So that is all I have for prepared slides, and we definitely have plenty of time. So I would love to take some time now to answer any lingering questions that people have. So I'm going to stop sharing. And I know we also wanted to take some time to take a picture at the end, so let's keep that in mind too. Wafa, you can tell us when we should stop and then get our picture.
Yes, sure. Thank you.
Mhm. Feel free to stick any other questions, and it could be from any of the stuff that we covered the entire 4 weeks. Feel free to stick any questions in the chat or raise your hand, and we can talk about it. Okay, well, if we don't have any additional questions, I think, Wafa, we can move to taking a picture maybe.
Yes, you're right. We can open your camera, please. Just a moment to adjust everything.
I'm sorry. Can I ask a question?
Oh,
Sorry.
yes.
Sure, yes. We have plenty of time.
Yeah. So there's this RAD54L gene. So when we were doing analysis in our lab setup, we saw that Franklin applies PVS1 criteria, whereas in ClinVer submissions, when we check for the null variants reported, there are no significant number of variants for it to classify PVS1 applicable. Like, so we want to know, like, you had mentioned that Franklin is a good website, like a resource which we could depend on for the clinicians in one of the workshops. So I want to know, like, why is there a discrepancy there, and is it anything about the updation, or so I want a clarity over that.
Yeah, so I would say if there is a lack of, I guess when I'm asking the question is loss of function and mechanism of disease, because I agree PVS1 can bump a variant to likely pathogenic very easily. I will look at a couple different resources. I'll do what you did and go to ClinVar and see, are there null or loss of function variants that have been submitted to ClinVar. I'll go to ClinGen and see, is there a curation for this gene, and and read the evidence summary and what types of variants are scored. And so in this case, I'd be looking for if they state anything about the mechanism of disease in their evidence summary and their curation. And then I would look and see what types of variants are they scoring, which is available in the genetic evidence section tab of the gene curation. And so that will further help me decide, am I thinking is loss of function a mechanism of disease? I will potentially go to the Gene Curation Coalition, look up the gene, and see what our other gene curation resources saying about this gene disease relationship. If I need to go further, I could go to OMIM and look at the variants table there. Are they listing any loss of function variants? And do I believe those loss of function variants? If I really want to go very far, I actually have developed a semi-quantitative rubric, I haven't talked about it here, for mechanism curation from the Gene Curation Coalition that is published on the ClinGen website. It's in draft, that you can score your curation and see if you get to an established loss of function, a likely loss of function, a suspected loss of function. Let me share my screen. I can show you where that is. So if you go to clinicalgenome.org and you go to working groups in the very bottom, and I can stick this in the chat too, working groups, and scroll to variant classification working group. You can then go to GenCC mechanism curation working group. The most tested one so far is actually the one that you're talking about, which is the draft loss of function mechanism of pathogenicity framework. And so if you were to click this, it is a framework that will tell you how to score curations to be able to get to this final classification of established, likely, suspected, or uncertain loss of function. So if you would really, if all of the other places that I suggested you look for this evidence don't convince you of the right answer, you could try using this framework. And so I will put in the chat where this is. And we have all the different frameworks here: gain of function, dominant negative, and not loss of function. The one that's referenced in the new guidelines right now is the loss of function one. So I would say that that's the most tested right now. So let me stick that in the chat so you have it. There.
Thank you.
I think we have another hand up. We see you unmuted, but we can't hear you.
Can you hear me?
Yes, yes, we can hear you now.
Yeah, thank you very much. I've learned a lot of things, so I would like to say thank you for the delighted, well-prepared presentation and the courses. This is a very, very great opportunity to me since I am from Tigray, and more genomic medicine is not yet started in our country, Ethiopia. We have, as I told you before, we have health science research, health institute. So we have also one PhD graduate from genomic medicine. So I think it is a great opportunity to me to start this course. My final question is, I think I cannot able to open the video camera. Would you assist me?
I don't know if you have any tips and tricks for that, Wafa. There's a little camera button at the top that you can click it and turn it on and off. Otherwise, it might just be your setup. Maybe there's permissions. It might not work.
He is allowed to share camera and mic, no problem.
Yeah, yeah. Yeah, so I think it's just the little camera button at the top. And if clicking that doesn't work, it might be, something with your computer. Okay, I have a question in the chat. If there's a pathogenic variant, autosomal recessive heterozygous, but with a genotype-phenotype compatibility, according to the literature, just one paper reported autosomal dominant mode of inheritance for the gene, do you consider it as a diagnostic variant? I would say, if I understand this question correctly, if you have multiple modes of inheritance potentially posed for a gene disease relationship, let's maybe say that you found a pathogenic variant and autosomal recessive condition that's just a carrier, potentially heterozygous carrier. But they're suggesting that there might be an autosomal dominant mode of inheritance also in the gene. Could you consider this a diagnostic finding? That's a tricky question. I've definitely had people reach out to me, and as you know, genes behave how genes behave. And so there can be some tricky situations where there's an autosomal dominant mode of inheritance and there's an autosomal recessive mode of inheritance. So I think you have to ask the question, what's the mechanism for the two different modes of inheritance? Is it something that you think should be semi-dominant, that, biallelic carriers, so those who have two variants are more severely affected? They might have more earlier onset conditions. So I'm thinking of like FH, familial hypercholesterolemia, with LDLR variants, where one variant is usually adult onset, and two variants is childhood onset, and that's something that you would want to report variants in the heterozygous case and in the homozygous or compound heterozygous case. There are some other conditions that For example, I'm thinking of GJB2 that has a dominant condition and a recessive condition. And the dominant condition is usually missense variance that might be a dominant negative effect. And the recessive condition is usually loss of function variance that both of them can have deafness, but the dominant condition will have a skin condition associated with it. And so sometimes you can tell based on the type of variance whether it would be dominant or recessive, because recessive is loss of function. Sometimes you're out of luck with missense variance, because if you have a missense variant, it hasn't been characterized, you don't really know. So I would say in a diagnostic setting, if that single variant might match the phenotype of the patient, I would maybe still report it and say, this could be a carrier, that we may not have found the second variant, that there has been evidence to show that dominant and recessive cases have been identified in the literature. But the thing you want to make sure of is there's a legitimate gene disease relationship with one mode of inheritance or both modes of inheritance, or kind of the spectrum of inheritance. You would never want to return something if there was only ever one autosomal dominant case ever, and you find one variant and say this could be autosomal dominant. In that case, you might want to say it's likely this is just a carrier variant, or we haven't found the second variant. But I would say if it matches the phenotype, I would probably report that variant and speak to the caveats. Okay, I see one hand up, so feel free to ask your question, and then I'll move back to the chat.
Yes, thank you so much, Dr. Marina, for the wonderful presentation and lecture about variant inoculation. It is a skill that we really needed so much, and for some of us, it came handy. I'm the national advocate for rare diseases in Uganda. We are preparing awareness campaigns. It's just today is 28 days away from the World Rare Disease Day. So this was a very wonderful and handy training. And I would want to call upon everyone here watching that rare disease is one of the areas where there is a very huge unmet gap. And I call upon everyone to use the skills we learned to impact rare disease in their places. And if anyone is doing research in rare diseases, we have a conference for rare diseases in Kampala in April. Maybe I'll leave my email there. They might email me, and then we could invite them to come and attend and impact rare diseases. Otherwise, thank you so much for the wonderful training. Bye-bye.
Great, thank you. I do this to try to make an impact, and I'm very passionate and excited about it. So I'm glad when others are passionate and excited about it. Okay, I have some other questions in the chat. So there's a question: with the assessment of clinical actionability, more emphasis is laid on systematic reviews and meta-analysis, but not case studies and case reports. Why? So I think in general, the actionability work in groups, and this is me speaking having not curated, so this is the best that I can say, is they really would like to see repeatability and statistical analysis. So a lot of times, while case reports can be really powerful, the reasons that they'll use a meta-analysis is because it's kind of combining all of the case reports in the literature. And systematic reviews will also do the same thing. And so oftentimes they will prioritize these things because they are, those reviews and analyses are looking at all of the evidence that's available instead of just single case level reports, where we know that oftentimes case level reports are really finding the most severe ascertainment bias. So those individuals that are very severely affected sometimes, and families that have really compelling evidence that might bias a bit more towards higher penetrance of conditions. At least that's what we've seen when we looked at genetic causes in biobanks, for example. And we are thinking about the actionability of those individuals who are unaffected, that you found a variant in one of these actionable conditions. So I think that's why they tend to look more at systematic reviews and meta-analyses. That being said, if those are not available, they will try to look at the literature that's available, knowing that it might be a rare disease, but it is because it's largely in a screening scenario, even though that's not necessarily the right word. It's really more for individuals who have diagnostic testing that you would find an additional variant that's unrelated to their phenotype. So it's a screening of sorts. So that's kind of why that they're looking at those meta-analyses or those systematic reviews. There's a question of how can we keep in contact? This discussion has been helpful. So everyone has my email address. I don't know if we have any other thoughts about how people could potentially keep in contact with members of this group. I think it's a great question that we could potentially brainstorm how people could keep in contact. We have like 189 participants, so it could be difficult to potentially be able to provide all of that information. But that's something to think about. I will say that that was a question one of the first weeks is, will people get PowerPoints? And so I will be sending after the fact, once I've grabbed them all, PDFs of similar presentations to all of the ones that you've seen in these recordings. I will say I did not give those presentations, so I can only provide the PowerPoint PDFs of things that I have that are quite similar. So I'll be sending those after the fact so that you have those for reference. So there was a question of, could I leave a link to my LinkedIn page for us to follow? Wafa did, I think, originally put that in the description. I can also grab it and stick it in the chat. But yes, feel free to follow me on LinkedIn for messaging. But you also have my email. Feel free to email me if you have future inquiries. Let me grab that now. Send it to you guys. Okay, here it is. There's my LinkedIn. Okay.
I think I sent them your LinkedIn the first email I sent
Yes,
to
yeah,
them.
it was in the first one too. So I stuck it in the chat again, but yeah, Wafa did provide that in the first one. There's another question, science question. Is there any evidence you use to assess epigenetic factors in shaping gene disease relationships, because the sequence alone cannot fully explain the phenotype or its effect on the severity of disease? It's a great question. I will say for gene disease relationships, most of the time we're not looking at epigenetics. We have started to move into a bit more complex disease or like regulatory genetics or epigenetics. So I think that's a still to be discovered section of ClinGen. I think we started a lot with rare disease and highly penetrant disease. And so keep an eye out on what ClinGen's working on. I would say the most common things in ClinGen right now that we're working on are more common variations. So the things like polygenic risk scores. Actionability is actually working on evaluating polygenic risk scores. We have complex disease working groups. Many of the groups that are working right now are those connected to autoimmunity or to immunity as far as complex disease. That's a one that we're working on. Pharmacogenomics, we have paired with CPIC and have created a new resource called ClinPGX, where the grants are kind of highly partnered now. So that's another space. So we are starting to explore these additional factors of regulation, but are not quite there yet. So then there are additional questions about creating groups for discussion, and maybe it's something that Wafa can think about a little bit, or I can think about to brainstorm a way, because we do have all the email addresses, a way to potentially come up with some sort of group to communicate after the fact, so that you're essentially a cohort that has worked together and can continue to discuss things. So maybe that's something we can think about. Mm. Okay, yeah. Thank you for volunteering to help create the community
Yes,
group.
thank you. So I will send to all participants and who want to participate in this community. I will ask Omar to add this participant in the community. Thank you, Omar.
Okay, I think we're a minute before the end, so let's try to grab that picture now so that we can have the nice picture together.
Yes.
So turn on your camera if you can.
Yes,
Love to
please.
see your smiling face.
I'm waiting. Yes, great. Yes. Turn on your camera. Thank you. Other one. This is the last one. Just a moment. Thank you. Thank you so much.
Great. Well,
Thank you
thank you,
so much
everyone.
for this
It has been a
opportunity.
true pleasure. It's been a true pleasure to interact with all of you, and I'm sure I'll see you in the genetics field at some point. You have my contact information. Feel free to reach out. I would love to stay connected with all of you. I'm so excited to be inspired by people who are really leaders in your field, in your country. So best of luck. I will send a PDF of all of these PowerPoints, and keep being passionate about genetics.
Thank you, Dr. Marina.
you very
Thank you,
much.
all participants. Yeah.
Thank you
Have a great
for
day.
all.
Thank
Bye
you,
bye.
Thank you,
Bye.
everyone.
Bye.
Thank you.
And Wafa Rashid, thank you very much.
Thank you very much.
Thank you, Dr. Zafe.
Thank you very much, everyone.
Thank you, thank you, thank you, thank you. We don't have anywhere to express
Thank
our
you so
feeling.
much, all.
Thank you very much.
Thank you.
Thank
Thank
you.
you. Bye-bye.
Thank you, Wafa
Bye-bye.
Rashid,
Bye.
for your coordination, Rashid. I think Wafa Rashid.
Yes, Wafa Rashid.
Thank you.
Yes.
Thank you
Thank
for
you.
your coordination. And it was a lovely week, by the way. I'm very, very happy.
Love you all.